Alright, in this segment, we're just going to talk about uh, PCR and how that works. So here we see PCR is replication in vitro, so in a test tube, um, in repeating rounds. So it's going to be a lot of cycles. We'll see in the pictures. Um, the idea here, or the trick here in PCR, is to replicate both of the double-stranded DNA at the same time, so you can focus in on one specific sequence or segment of the DNA you're looking at. Um, and so you have two primers, one for the 5 prime to 3 prime, the other one that's on the 3 prime to 5 prime. You'll see pictures of this too, so it's more clear in a little bit. And the primers are 20 base pairs long, and you can make sequences, you can PCR sequences, or PCR meaning you can exponentially enlarge the DNA from using PCR. So the PCR fragment can be from 20 base pairs to 10,000 base pairs, but like if the base pairs are bigger, the less yield you're gonna get. And you need heat stable DNA polymerase to be able to use PCR, because we increase the temperature to 90 degrees Celsius, and most proteins would denature that, so we need a special protein for that. So we'll see later on that how PCR is used in sequencing DNA also, and genotyping. All the different applications of PCR. So the first is DNA sequencing, second is genotyping. So we're going to use SNPs, single nucleotide polymorphism, which is just a single nucleotide difference. We can see what I mean by SNPs over here. So we have we have these two DNA fragments, AAGCTA and then AAGCTTA. So the C is replaced by a T, so it's a single nucleotide change. That's SNPs, and and then replacement of southern blotting and microsatellites, which are mostly CA repeats. And we'll talk about those more with the pictures. And we can find out if there's HIV infection or any other viral infection using PCR, because we can do reverse transcription, get the cDNA, and then do PCR on that, and then use the gel for that. And also you could do forensic genotyping for like Ramsey and stuff like that, to see for like the variable regions in the human genome to identify a person. And then we can measure how much mRNA is there in the cells by using reverse transcription to amplify it. Okay, so the steps in PCR with the pictures. So this is the whole genome, and we want to just this little segment, because you can this whole genome could be up to 3 billion pairs spaces long, and you only can do up to maybe 10,000. So maybe you only want to do like 300, or somewhere between 20 and 10,000. So that's what's going to be over here. We have 3 prime to 5 prime, 5 prime to 3 prime, and It's going to be synthesized. The new strand is going to be synthesized 5' to 3', and it's being read 3' to 5'. So you heat it to 92 degrees to denature the DNA, and you have two separate strands, and then you add the primers. So this primer is binds to this part over here, same thing over here, and then you add polymerase, or the thermostable DNA polymerase from the bacteria that you get from Yellowstone, where and that heat stable DNA polymerase is stable because of the disulfide bonds. It has an increased number of disulfide bonds, and disulfide bonds are covalent bonds which won't be affected by heat. They will after really high temperatures, but 9 degrees is okay. So after the first run through, you get, so this part's going to get cut off, and this part's going to get cut off, but you have this extra and this extra after the first cycle of the PCR. Usually just put the program the machine to like 20 cycles at the same time. So then you have go like that and goes like that, and then after, so once you have this, you have, and then you have that, and then you separate it, and this is going to be the right size, and that's going to be still bigger, and then eventually you get just this one's going to be right size, that's going to be too long, and this one's going to be the right size, this one's going to be too long, but these ones are going to increase linearly, and this one's is going to grow exponentially. So in the end, you're going to have a lot more of the gene of interest and a lot less of the extra base pairs one. So the target sequence will be amplified 10 to the 6,000, 10 to the 6 fold. So it's going to be a lot more. And if you understand this part, you should be able to understand this question over here. We'll come back to that question later. And so here is just another diagram of PCR amplification. So you have the cells, and then you isolate the DNA, and then that's the section to be cloned. So then you have primer here, gets that, primer here, gets that, and then you'll end up with just that part. And then in mRNA, first you have to get the cDNA. To get the cDNA, you use reverse transcriptase, and you put the primers in, they'll anneal to the RNA, and then you use reverse transcriptase, and it'll make a DNA. Take that DNA, and then you just run a PCR reaction with that. So you just add one primer, and it's going to attach to all these ones, and it's just going to keep on growing them. And then that's it for the reverse transcriptase. So let's say you want to know if a person has HIV. Take some of their blood, and then you have primers for the primers to cut the sequence where the cut the sequence where you, so first you have to know the sequence for the HIV virus. So then you can design the primers, and then you put the primers here. So that's, let's say that's the HIV RNA. So you put the RNA HIV sequence, put that there, and then use reverse transcriptase to elongate it. So we're assuming the person has HIV, right? So you put the primer, it'll anneal over here, and then you can take you can take like some cells and then just look for the RNA in that cell and then just isolate the RNA, and then you would do this whole thing. And then you do gel electrophoresis, and then if it shows up a band and it's expected size, then you know the person has HIV. Or you can just run regular PCR and look for, see if some of the cells have the HIV sequence incorporated into it. And over here, this graph is showing how you can tell which of the products you have more. So you have mRNA A and mRNA B. So the mRNA A is less because, no, it's more because it took off. It started growing more quicker. Because, like, you run through the cycles and it grows exponentially after a certain point. After, let's say this one started growing exponentially after 15 cycles, and this one at 20 cycles. So you start off more of this. So the conclusion is mRNA A is more abundant to begin with. And then microsatellites could be different in the homologous chromosomes. So you have a primer, and then the VNTR, the variable number tandem repeat, just means the microsatellite. It's another name for microsatellites. And you have the primers, and then you extend it, and then you check it on gel to make sure you got the right one. And that's how you check for microsatellites. Not sure what else you need to know about that. And this question over here. So question's asking, what primers do you need to copy this segment? So let's say there's a lot of this. So there's a whole bunch of these in between these two. So we're gonna have to make a primer that goes this way and a primer that goes this way. So we had CAG dot dot CAG TCC and CCG and CCG, and we got 5' on this side and 3' on this side. So they're asking us what would be the primers. So then what you would do is so let's write the sequence in the complementary of this. So it will be 3' G T C A G G dot 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 G T C dot 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 G G C G G C 5'. So that would be this would be the thing, and then you separate them by annealing, by denaturing them, and then you would need a primer here. So it would go from 5' to 3'. It will be synthesized 5' to 3'. So the primer would need to be C, G, G, C, G, G, C, G, G. G, G, C, G, G, C. And this side would be 5 prime, and this side would be 3 prime. So that would be one primer. The other primer would be, because the primer is gonna be synthesized 5 prime to 3 prime, so that's why for this it should be on this side. And then over here the primer would be C, A, G, T, C, C, dot dot dot. That would be the primer, 3 prime. So the two primers are this one and this one. These are the two primers. And then if you look at the question and you see, so we're looking for so we always write it from 5 prime to 3 prime, so we'll have to flip this one. So C, G, G, C, G, G. So I just flip this around. So it's gonna be 5 prime, 3 prime. So these two would be the primers. Oh man. Finish this off later.